All right, so on to the fats that we find in the membrane before we talk about some of these proteins and carbohydrates and things. So the membrane fats, the types of phospholipids that are actually in there, the fatty acid chains, can be different, can change from season to season, for example. And what we're looking here is at the percent of the unsaturated fatty acid chains. Remember, saturated means it's got all sorts of hydrogens on it, all single bonds. Unsaturated means there are some double bonds in those fatty acids, and so they don't have all the hydrogens that are possible. So what does that mean? Well, if we've got an unsaturated, get rid of some of my drawings here, we've got an unsaturated fatty acid tail right here. It gets little kinks in it. Okay, so all these fatty acid tails in here have double bonds in them, and that makes them kind of, crooked. Whereas if they're all saturated, in other words, they're all single bonds, so they are full of hydrogens, they're nice and straight. All right? And what happens to the membrane when we change those fatty acid tails is it can get more fluid or more viscous, which means like more jelly-like. Okay, so if the membrane needs to get a little bit more fluid, the cell is going to start building phospholipids that have unsaturated fatty acid tails. If the membrane needs to get a little bit more jelly-like, it's going to build phospholipids that have more saturated fatty acid chains. Okay, so the cell will adapt the composition of its phospholipids based on if it needs to be more fluid or if it needs to be more jelly-like. And why would a cell need to be more fluid or jelly-like? Hopefully is the question you're asking yourself now. And the answer has to do with temperature. Okay, so if the temperatures are getting colder, like for example in plants like winter wheat that grow in very cold temperatures, the fats are going to start to get more solid. Think about butter, right? You put butter in the refrigerator, and as it gets colder, it gets harder and harder and harder. Well, you want your membrane to be a little more fluid than that. So as it gets colder, organisms, again like this winter wheat, are going to be putting more of these unsaturated fats into their membrane so that their membrane can stay more fluid. Okay, we don't want a hard, solid membrane as the cells get colder and colder and colder. You still need to allow it to be fluid and to move and to squish and things like that. So again, as it gets colder, it's going to get more unsaturated fats in the membrane. Okay? Hopefully that makes sense. So as we see, as I said, as it gets colder, the seasons change. You're going to see an increase in the percentage of those unsaturated or fluid-like fatty acids in autumn. And then, of course, a decrease in the summer and the spring. Cholesterol in the membrane can also serve this function. So cholesterol can control how viscous or fluid the membrane is as well. So the cell has kind of two ways of doing that. And that's what that cholesterol that we were looking at earlier, the little yellow hexagons or whatever they were embedded in the fats, those are indicating that that can happen as well, to change the fluidity of the membrane. Okay, now on to our proteins. The first type of protein are the peripheral proteins, which are what they sound like. If something's on the periphery, it's on the edge of a structure. Okay, so in this picture, our peripheral proteins are going to be those that are only on one side or the other. In this case, I see a peripheral protein sticking up right here and right here. The other purple guy, oh, and then there's one down here. The other purple guy is not a peripheral protein because it goes all the way through. All right, so that's the difference we're looking at. Periphery is just on the edge. They are loosely bound to just the surface of the membrane. So again, they don't go all the way through. They often act as identity markers for the cell, or what we call antigens, and we'll look at those a little bit later about how that works and what that does. Our integral proteins are the ones that go through. So if something is integrated into something else, that means it's a part of it. So this big guy right here is an integral protein. It goes all the way through the cell membrane. So as I said, they penetrate through the lipid bilayer. Often they are transport proteins for moving things through the membrane, so they're like little tunnels or channels. And they could also be a pump as well, to pump or force substances through the membrane. Okay, so now we can further divide these types of proteins into their different functions or what they do. So you can see the setup here of how this is arranged is we've got the blue area of each picture is the outside of the cell. The yellow area of each picture is the inside of the cell. And then hopefully by now this plasma membrane looks familiar to you. And then each of these purple guys is a different type of protein that can be embedded in the membrane that does a different job. So we're talking about what each of these actually does. So the first one, as we go from left to right, is called a transporter protein. I think that's pretty clear based on its name what it does, and the picture shows you that as well. It's moving something into the cell. Transporter proteins could also move substances out of the cell, but it gets it through the cell membrane. The second one acts as an enzyme. So this protein is actually an enzyme that's embedded in the cell membrane that has an active site. Hopefully you can see that. Just circle it for you here. See this little shape cut out right here? There's its active site. It works on a substrate that fits into the active site and changes it into a different product. So that's embedded right there on the membrane. So the protein is an enzyme. The third type is a surface receptor. So what's happening in this case is we have some sort of little molecule outside of the cell that possibly another cell has sent to talk to this particular cell. So some sort of molecule that gets attached to the outside of the protein. And when that happens, it actually creates a chemical reaction within the protein that builds some sort of product inside the cell. So it's not like this little molecule out here travels through the membrane. It just binds to the protein, changes the protein, and thereby creates some sort of action inside the cell. Okay, the fourth one, cell surface identity marker. That's the one we were talking about earlier that I said I'd get back to. This is that antigen, and I think this does come up one more time later on too. Okay, so you can see this as a glycoprotein. So we've got our carbohydrate here, we've got our protein here, and these carbohydrates are specific to certain cells. So I have different carbohydrates on my cells than you do on your cells. So if you were to transfer one of your organs to my body, for example, my body would not recognize that organ as one of mine. It would see it as your organ instead because you have different markers on your cells. So transplant patients have to take certain medications so that doesn't happen to them, so they don't start to attack the actual cells that they're supposed to be keeping in their bodies to help them, the whatever organ has been transferred into their body. This also, as you can imagine, has something to do with your immune response. So when a bacteria comes into your cell, it doesn't have the right surface markers, and so your body knows to attack it. All right, so these are attached to proteins in this case, which makes them part of the cell membrane. All right, our fifth type is to make cells stick together, so cell adhesion. So you can see we've got two different cell membranes here. All right, here's one here, and here's one here, and each of them has a protein in it. So let's just highlight those proteins. Here's a protein, here's a protein, and right in the middle they stick together. Now what kind of cells would need to stick together in sheets? Well, your skin cells, for example. Okay, your skin cells have proteins in their membranes to help hold one skin cell to the next. Okay, that's called cell-to-cell adhesion. And then finally, the last one, attachment to the cytoskeleton. As I said, some microfilaments are actually attached to the cell membrane, and you can see how this protein down here, right there, is working together with the bigger one to actually hold those microfilaments, those parts of the cytoskeleton, in place, to anchor them where they need to be. Okay, and again, this is just looking at this a little bit more closely, but remember the carbohydrate has a role in the cell membrane as well. We're just talking about all sorts of proteins, but we brought up these special glycoproteins, which have carbohydrates attached to them for cell-to-cell recognition. So don't forget, we've got lipids, we've got proteins, and we've got carbohydrates as part of the cell membrane. And we discussed how antigens, or markers on the surface of your cells, can help your body to identify which cells are yours and which cells are maybe bacteria cells, for example.